My name is Michal Linial, and today I'll be briefly discussing microRNA prediction. This work was led by Dan Ofer, who is probably sitting in Israel and listen to us. So let's get going. What's microRNA is all about? Those are really the bodyguard of cell identity, and they do so by binding directly to the 3' UTR of any of transcript. Once this is done, there are two events that occurs. Translation is inhibited, and the stability of the transcript is reduced, is degraded. So in terms of numbers, if you look at human, we have 2,600 functional microRNA. Those are very small, 22 nucleotide unit. However, it is known for over 20 years that there are about 60% of the transcript that are regulated by microRNA. Nevertheless, this number has not changed despite the 20 years, as you can see in the graph, of changing the field from 0 to what we have as of now. So what this number came from? Two things to mention is that this estimate has not been changed, but all over, many of those microRNA might be not real. So having this, what we know about this target pairing issue that I mentioned, we know that we have hundreds of tools, algorithmic tools for microRNA target, too many of them, with very little embarrassing non-overlapping between them. And in recent years, we have hundreds of experimental tools like CLIP and so on that are too specific. So actually, there are too many false positive. And the question is, what can we do? In order to address it differently, we decided to do a machine learning, supervised machine learning model, quite standard one. But for asking this, you have to understand what is the input and how to validate your result. And indeed, the input of microRNA comes with different layers, such as molecular layer, cellular layers, network, and also evolution. So just to be very brief on the molecular level, this is just example of a 3' UTR of a gene. And you can see that there are tens or tens of microRNA binds to it, but the different colors means different affinity and different confidence. In terms of the number of the cellular level, we know that microRNA can be very different between the different cells and different tissue, and they're very different in expression in different tissue, as you can see here. So in terms of, I would say, evolution, unfortunately, we know quite a lot about the human and very few, almost nothing, about other organisms besides mouse that you can see that the rest are really very negligible. So the question we would like to ask is, can we decide which gene or proteins is a microRNA target? And the idea, just the rationale, is that coding region of most genes are under strong selection, as we know, and potentially they capture hidden information on essentiality under a microRNA regulation. In order to do the test, we took the state-of-the-art miR base, which is relying on many literature, and there are over 2 million of such interaction. But we took only the strong one, the one that we feel strong with. Then we took everything that we know about protein that we heard, which is UniProt, SwissProt, amino acids, global information, functional annotation, Go, cellular, and so on and so forth. And we engineered many features like we heard in previous talk. Altogether, for each model, we included about 20 million candidate feature from all combination per organism, which include everything that you can think or not think even. And we took the 300 most informative feature to say what to build the model, and immediately you can see that the model for the human is getting an AUC of 77%, the mouse a little bit less, C. elegans also 80%, and so on and so forth. So quite impressive AUC. Remember that we only included protein information, which has nothing to do with the transcript itself. This is just the AUC. You can see this 78%. that I showed you, and I don't have time to go into the different model, but the model that only use, let's say, the protein itself without the annotation is less than with annotation and so on. Most importantly, we were able to find what are the impact feature on the human model, and all those on left were positive, I mean, increased the model. Those on the right decreased the model, and just I marked, for example, the length of the protein. If they are shorter, they are less likely to be regulated and so on. But of course, this is not enough. The last part that I want to mention is to go into evolution and try to understand what does it mean in terms of protein families. Now, on the left side, you see a paralog. What's paralog? Those are, of course, after duplication within an organism. And we decided to take the big family, like GPCR or RAB family with the many different proteins, and to ask how they behave in terms of regulation. And when we do this, you see the 100% regulation from 0 to 100. And to our not surprised, but we were very delighted to see that some families are more likely to be regulated than other families. For example, the kinome, the kinases, are those that are highly regulated, including the RAB proteins or the signal transduction RAB protein. However, the histone, the peptidase, the other GPCR, they were much less likely. Actually, when we looked at the paralog, we were surprised to see that this regulation does not hold at the protein level, but only at the family level, going back to the rationale that I mentioned on evolution. And this is two examples of two families of the GPCR that you can see that this check mark are basically random in this phylogenetic tree. The last thing we say that we can transfer from one organism to the other. So we train on organism X and we predict on organism Y, and we can do it pretty successfully from human to mouse and so on. And just to say why is it important, we can go into organisms such as zebrafish and find genes from human known diseases that are relevant to this. And to wrap up, I think I showed you, so I don't have even to summarize, but it's important also for gene and disease and so on. And thank you very much. I tried to catch the 2 minutes that we lost. So I did it fast.
The online question was, were all these different species models trained from scratch, or were you able to leverage transfer learning to train one model and then restrain it for other species?
Yeah, it's a very good question. As I mentioned in the beginning, there are only mouse and human that are good model. So we learned mouse and human, and actually we were able to transfer, and that was very nice because even, and as I mentioned in the zebrafish or fly, that they do have small number of microRNA known, we were able to extend it. So for example, the C. elegans has only 2% experimental data, and we were able to get 70% success rate. So it was pretty good in transfer.